Regulation of gene expression is a way for cells to be efficient with their energy. Through regulation, a cell can turn on gene expression for proteins that are needed and turn off gene expression for proteins that are not needed. Regulation of gene expression occurs at the level of transcription, so cells do not make mRNA that is not needed. In prokaryotic cells, genes are located in operons. An operon is a cluster of related genes that are all under the control of one promoter and are transcribed together to form a polycistronic mRNA. There are three main types of transcriptional regulation. These types include negative control, positive control, and global control systems. Negative control of transcription is defined as a mechanism that stops transcription through a repressor protein. The repressor protein binds to the operator and transcription is turned off. There are two types of negative control, repression and induction. Repression occurs when there is too much of a product in the cell, so the pathway is blocked from making more. In induction, an enzyme is only made when its substrate is present. Positive control of transcription is defined as a mechanism that occurs through an activator protein that activates the binding of RNA polymerase to DNA. The activator protein binds to the activator binding site, and transcription is turned on. In global control systems, multiple genes and operons all respond in coordination to a change in the environment. Now that we have defined the types of regulation, it is important to define the different regulatory molecules that may be present in these mechanisms. One regulatory molecule is an allosteric protein. Allosteric proteins have two binding sites, an active site to bind substrates and an effector site that binds effectors. Two types of allosteric proteins are repressor proteins and activator proteins. A repressor protein binds to the operator and blocks RNA polymerase from binding, which results in transcription being turned off. Activator proteins bind to the activator site, resulting in increased binding of RNA polymerase and transcription is turned on. There are also two main types of effector molecules. Inducers are molecules that bind to repressors and transcription is turned on. Co-repressors bind to repressors, and as a result, transcription is turned off. In Mycobacterium tuberculosis, there are many examples of regulation that occurs. One example of a global control system can be found in the RV2390C RPFD operon. The transcription of the genes in this operon is affected by the changes in the surrounding environment. It has been shown that both an increase in chloride ions and an acidic pH increase the transcription of the genes in the operon. The greatest rate of transcription is seen in an environment that has both an acidic pH and a high level of chloride. The chloride acts as an inducer to turn on transcription. Mycobacterium tuberculosis contains a number of transcription factors called sigma factors. The sigma factors control the stress response in the cells. Different sigma factors respond to different types of stress, both in the cell and in the environment. When the cell is in a stressful environment, sigma factors act as activators to activate the transcription of genes that will help the cell survive in the harsh conditions. When a specific sigma factor is not needed to help the cell survive, an anti-sigma factor will bind to the sigma factor. This turns transcription off until the cell is once again placed in a specific situation where the genes are needed. These are just some examples of how regulation of gene expression is used in Mycobacterium tuberculosis in order to conserve energy and allow the organism to have the best chance at survival.